Today we are going to discuss a new topic of bacteriology, that is bacilli. And this topic we will study in different headings. Number one, we will study the general characteristics of Bacillus, and then species, Bacillus Anthracis, morphology, staining procedures, cultural characteristics, biochemical reactions, resistance, antigen structure, virulence, epidemiology, clinical manifestations of anthrax, and in last we will discuss about the laboratory diagnosis. So we will discuss one by one. First we try to understand the general characteristics of this Bacillus. The genus Bacillus consists of obligate aerobic gram-positive spore-forming rods, and they are generally motile and peritrochus flagella. They are motile and peritrochus flagella. And non-capsulated, and we will see that anthrax bacillus, which is non-motile otherwise and capsulated. Otherwise all bacilli or bacteria are capsulated, and the most of bacilli are catalase and most produce acid but not gas from glucose. That we will see in biochemical reactions. And the genus includes psychrophilic, mesophilic and thermophilic species. The maximum temperature for vegetative growth ranging from about 25 degrees centigrade to above 75 degrees centigrade, and minimum from about 5 degrees centigrade. 25 to 75 और मिनिमम देखेंगे तो हमारा 5 डिग्री सेंटीग्रेड तक भी हो सकता है, 5 to 45 डिग्री सेंटीग्रेड. अगर ये दो हो गए 25 डिग्री सेंटीग्रेड to 75 डिग्री सेंटीग्रेड and 5 डिग्री सेंटीग्रेड to 45 degree centigrade. These are the two ranges, and their spores are ubiquitous. Ubiquitous means they are found in general. They may be found in soil, in dust, in water, as well as in air. So we call that their spores are ubiquitous. Ubiquitous. This is all about the general characteristics of bacilli. Now we will see about the species. That despite the subdivision of Bacillus, there are more than 50 species. More than 50 species in this genus. And fortunately, the species that are of medical interest are relatively limited. And we will study about the Bacillus Anthracis. Okay, we will study about this Bacillus Anthracis, and this organism is responsible for disease. This is known as anthrax, and the most important member of this genus. And second one is Bacillus cereus. Bacillus cereus. This is the second bacilli, and it is commonly implicated in episode of food poisoning and responsible for opportunistic infections. मतलब इनको मौका मिल गया तो ये इन्फेक्शन कर देंगे, अदरवाइज कोई बात नहीं. सो नाउ कम टू अवर दिस Bacillus Anthracis. Now this is the topic for 2 days, and it is a causative agent of important zoonotic disease, and that is called as anthrax. The disease primarily of animals, and man gets infected secondarily. And it is also gained importance recently because of its ability to be used as biological weapons, just like corona. And it was first pathogenetic bacterium to observe under the microscope, and that is observed in 1849. What Koch, as you all aware about the Koch, who gave us the Koch postulate, and he isolated this bacilli in pure culture, and in 1876. And Pasteur was the man who prepared an attenuated vaccine by using this Bacillus Anthracis in 1881. So this is all about the history. And one more thing is Nobel Prize winner Mechnikov studied the virulency and attenuated strains of V. anthracis in his pioneer work of phagocytosis. जब वो phagocytosis पे वर्क कर रहे थे तब उन्होंने इसकी virulency को देखा था और समझा था. ठीक है ना. दिस इज सिंपल हिस्ट्री ऑफ Bacillus Anthracis. Now we will go for morphology. In morphology we study the size and shape of bacteria. So it is the largest of pathogenic bacteria, and the diameter and breadth is 3 to 10 micrometer. 3 to 10 micrometer, और इसकी जो breadth होगी that will be the 1 to 1.6 micrometer. That's why we are talking that this is the largest pathogenic bacteria. And it is gram positive, number 1, gram positive, non-acid fast bacilli, non-acid fast bacilli, non-motile. These are the characteristics of this bacilli, and it is found in rectangular shape and arranged in filamentous chains in culture. एंड हम अगर इसका कैप्सूल देखेंगे, क्योंकि ये कैप्सूलेटेड होती है, तो इट हैज पॉलीपेप्टाइड कैप्सूल. कैप्सूल विल बी पॉलीपेप्टाइड. जबकि दूसरों में किसका होता है, दूसरे बैक्टीरिया में देखेंगे तो वहाँ पॉलीसैकेराइड होता है. Others. In case of this, it is of polypeptide capsule and the entire chain is covered by this. ठीक है. And this is made up of polyglutamate. Polyglutamate. And this capsulated bacilli are often gave a appearance of boxcar. Boxcar. This is like a chain and it seems like a boxcar. And the spores are oval. जब हम इसमें कोई ये हमारे सेल के अंदर ओवल ये होगा, that will be the oval. And refractile, central in position, and of the same diameter as the bacillus, so that they do not cause bulging of vegetative cell. And it cultures, the bacilli are arranged end to end in long chains. End to end, मतलब एक छोर से दूसरा छोर, एक छोर से दूसरा छोर. The ends of the bacilli are tuberculated or often concave and somewhat swollen, so that a chain of bacilli present a bamboo stick appearance. बांस का आकार जैसा हमको दिखाई देगा, and that the end will be the concave. And the bacilli are found singly, in pairs, or in chains and tissues. This is the morphology of this bacilli. Now we will study about the staining processes. In staining we use the McFadden's reaction, in which we take a smear that contains Bacillus Anthracis and stain with polychrome methylene blue. Polychrome methylene blue. Polychrome methylene blue. For a few seconds and examine under the microscope, then we see that a amorphous purplish material is around the blue bacilli, है ना? जब हम माइक्रोस्कोप में देखेंगे तो हमारे जो bacilli है उसके चारों तरफ हमको एक amorphous purplish material दिखाई देगा, and this is the characteristic material of or appearance of this bacilli. And while we stain under the Gimsas stain, then the purple bacilli with red capsule is seen. यहाँ पे कैप्सूल रेड होगा और बेसिलस हमारा पर्पल होगा, वहाँ पे हमारा ब्लू होगा और एमोर्फस जो मटेरियल है पर्पलिश कलर का चारों तरफ दिखाई देगा इन मैकफार्डीन्स. Then we see in culture bamboo stick appearance, in which we see the end-to-end arranged long chains of bacilli in clusters. एक से एक वो एक bacilli जुड़ा हुआ होगा जो हमें bamboo-like appearance दे रहा होगा. And we will see the now we will study the culture characteristics. After studying we see the cultural characteristics. हमने कल्चर में क्या ग्रुप या तो उसके क्या characteristics दिखाई देंगे. They are aerobic, facultative anaerobes. Optimum temperature for their growth is 37 degrees centigrade and the range will be the 12 to 45 degrees centigrade. The optimum pH growth for growth is 7.4 and the optimum temperature for sporulation is 25 to 30 degrees centigrade. And for germination of spores requires a fresh nutrient and aerobic conditions, and the growth and the good growth occurs on ordinary media. So when we use the nutrient agar, then we will see the colonies will be irregularly round, or they will be seen in 2 to 3 millimeter nutrient agar and a nutrient agar. Okay, and the colonies will be seen raised, dull, opaque, grayish white with frosted glass appearance, जैसे कोई धुंध जमी हुई हो ग्लास के ऊपर. And under the low microscope, the edge of the colony is composed of long interlacing chains of bacilli, and that resembles locks of matted hair. जैसे बालों का गुच्छा बन गया हो, वो इस तरह से दिखाई देंगे. And this is called the medusa head appearance. Medusa head appearance. जैसे बालों का गुच्छा आपस में उलझ जाता है, वो that will be seen, and it is known as medusa head appearance. Okay, and this is seen under the nutrient agar. And now if we culture on blood agar, then the colonies will be non-hemolytic, though occasional stains produce a narrow zone of hemolysis. And when we done in broth, the growth occurs as a floccular deposits with little or no turbidity. And when we do the culture in a gelatin step culture, gelatin step culture, in which a characteristic inverted thread tree appearance is seen, with slow liquefaction commencing from the top. Fine filaments of growth develop laterally along the line inoculum, जहाँ हम inoculate करते हैं वहाँ से, and the growth near to the surface of the medium is the longest and then progressively shorter when there is less oxygen resembling a inverted thread tree. नीचे ऑक्सीजन कम होगी, ऊपर ऑक्सीजन ठीक होगी, तो ये हमारा ऐसा लगेगा कि ट्री को इन्वर्ट कर दिया है. ठीक है ना? This is the gelatin step culture. And now we will talk about the next selective medium, and that is PLET medium, and P for polymyxin, L for lysozyme, and E and T for ethylene diamine tetraacetic acid, that is also known as EDTA, and it is used as anticoagulant especially in laboratories. And thallose acetate added to heart infusion agar, and this helps in isolation of Bacillus Anthracis from mixtures containing other spore bacilli. जो दूसरे bacilli होते हैं उनमें से इसको निकालने के लिए ये यूज होता है. And now we will talk about the biochemical reactions. And this ferments glucose, maltose, sucrose and dextrin with the production of acid, and there will be no gas, and it reduces the nitrates to nitrites, and the catalyst is formed positive. This is all about the biochemical reactions, and next remaining portion we will study or discuss in next lecture. So thank you.